Okay, welcome to the review for topic 6. I'm going to go quickly through the first few slides and everything. Each slide has the question and the answer from the study guide, and I also posted a copy of the answer key on Google Classroom as well. So let's get started. So we talked about this one in class today. It says outline the steps of blood clotting in the human body. Just an overview. There are two key components to blood clotting, platelets and insoluble fibrin. So the coagulation cascade, remember coagulation is to like group together or gel up. And then a cascade is like a domino effect. So coagulation cascade is triggered when clotting factors are released from damaged cells and platelets. I would say that this part right here, like where it's released from damaged cells and platelets, like that part's super important. Clotting factors trigger conversion of prothrombin to thrombin. Thrombin triggers conversion of fibrinogen, which is soluble, to fibrin, which is insoluble. And then the fibrin forms mesh around the sticky plug of platelets to trap blood cells and form clots. Question number 2, what are viruses and why don't antibiotics work against them? So just as a reminder, viruses are non-living pathogens. And the way that antibiotics work is that they target prokaryotic metabolism, which remember metabolism is like cellular function. Because viruses don't have metabolism, because they're non-living, antibiotics won't work. All right, so this is the label diagram of the heart. You can take a picture of this or pause the video in order to copy down the stuff that we did. So I'm just going to leave that there for a moment. All right, the steps of a heartbeat, including systole and diastole. This is one of the last things we talked about today. The heartbeat is myogenic. That means it's initiated by heart muscle cells and not brain cells. The SA node, remember that's sinoatrial, that is the pacemaker. That's what initiates the heartbeat. It sets the pace for your heartbeat. So the SA node is going to stimulate contraction of the heart muscle, which causes the atria to contract, which then stimulates the atrioventricular node. The AV node then stimulates fibers in the ventricular walls, causing ventricular contractions. So systole is periods of contraction, and diastole are periods of relaxation. So these things right here, I kind of, put that on my paper, that these two kind of statements here are about systole. I would say that the most important thing for you to review from these statements are what's happening with the valves during systole and during diastole down here as well. Okay, so this is one we already talked about. As things contract, they also relax. Valves are open and closed. So just take a look at that. All right, so what is the ileum and what kind of tissues are found there? So the ileum is part of the small intestine. You can see this picture right here. I did not put this on the answer key, but I put it here so that we could look at it. So you have 3 parts to the small intestine. You have the duodenum or the duodenum, and then you have the jejunum and the ileum, and you can see those in the picture. It's part of the small intestine, and the tissues in the small intestine include something called a mucosa, which is a highly folded inner layer. Think like mucus, right? Then you've got smooth muscles. You've got serosa, which is a protective outer layer. And you do have villi up in the small intestine because that's what helps absorb nutrients. What types of tissues are found in the stomach? Mucosa and smooth muscle. What area of the heart has the highest blood pressure? Okay, so it's the left ventricle. It's responsible for pumping the oxygen-rich blood to the body through the aorta. So the left ventricle, because it has such a high blood pressure, has thicker muscular walls. And this is just right here. This is just a reminder of what an artery is. Arteries, in general, convey blood at high pressure from the ventricles. So this is just an extra statement I put down here at the bottom about the structure in order for arteries to accomplish the task of moving blood at a high pressure. So they've got a narrow lumen, and remember the lumen is like the tunnel in the middle, if you will. So they've got a narrow lumen to help maintain that high blood pressure. They have a thick wall containing an outer layer of collagen to prevent the artery from rupturing. And then they have an inner layer of muscle and elastic fibers. to help with the flow. This part is not like the structure, specialized structure is not as important to study, but you do need to know which area of the heart would have the highest blood pressure. All right, here are the risk factors for developing lung cancer. So we wrote these down today, but I like this picture because it has some examples, right? So radiation like x-rays, aging, pollution like smog, environment like the radon levels, diseases such as COPD, genetics and family history, occupation like miners breathing in different chemicals and things like that. Asbestos, we talked about that it's found in a lot of older homes. And tobacco smoking and secondhand smoke. These are all big risk factors for developing lung cancer. And as long as you're able to recognize a few of them, you should be okay. All right, the difference between type 1 and type 2 pneumocytes in both structure and function. So we did talk about this today. Type 1 is squamous, which means they're flattened and they're thin, and they're involved in the gas exchange. Type 2 are cuboidal and produce pulmonary surfactant. So remember, surfactant is kind of like a lubricant of sorts, and it helps keep everything nice and smooth. And we'll talk about that picture down there in a minute. But over here, we have a high magnification. That's this picture. We have a high magnification of what the pneumocytes look like. So the type 1, it's this like really flat squamous cell, so it's flattened, super thin. And then we have the type 2, and you can tell how much bigger it is width-wise than the type 1 pneumocytes. And the type 2 is, again, creates that surfactant. So down here, the role of surfactant in regulating surface tension. So without the surfactant, alveoli 1 and 2 have equal surface tension. 1 has a higher pressure, so this would be 1, and this is 2. 1 has a higher pressure due to a smaller radius, and 1 is more likely to collapse and be harder to inflate. Now, when we have the surfactant, that's the second part of this picture, 1 has, again, 1 and 2, has less surface tension because it's got more surfactant per area. 1 and 2 have equal pressure due to the surfactant, and number 1 will inflate at a faster rate than 2 until they're equal in size. Okay. Okay, number 10. Let's talk about this one. Outline the steps of inspiration using the following words. So inspiration, just so you know, and we don't mean inspiration like, oh, I feel so inspired. This is air moving into the lungs. How that happens or what it's due to is pressure in the chest being less than the atmospheric pressure. So here's your little statement right here that uses those words. During inspiration, so during breathing, when you're breathing in, the diaphragm is this guy right there. It's a muscle underneath the lungs, helps move the lungs up and down. So the diaphragm is going to contract, and when it contracts, it kind of flattens. What that's going to do is it's increasing volume by moving the rib cage upwards and outwards, decreasing the pressure inside the lungs. So if you take a nice deep breath and you kind of think about what your body is doing when you take a nice deep breath, your rib cage is moving up and kind of out. And what happens is you're increasing the volume of your lungs by breathing in. And when you do that, it decreases the pressure inside of the lungs. This is supposed to be a nice, good little GIF, but it's not working. So, okay, this one right here, number 11. What does it mean for nerve fibers to be myelinated? What does myelination of these nerve fibers cause? Okay, so the first thing is this picture right here is a picture of an axon, which is a nerve cell. It's got the parts labeled and everything like that. But their axons, for the most part, are covered in something called myelin. And that is a fatty white substance that acts as an insulating layer. So in this picture, we call it, when it's all together, we call it the myelin sheath. And that's these like yellow little like sections that you see in the picture. So the main purpose of the myelin sheath is to increase the speed of electrical transmissions, or what we call saltatory transmission. And that's going to be the next question about like number 12 is what is saltatory transmission? Okay. But while we're here, I want to point out a couple of parts. So we have the myelin sheath. These little guys on the inside are called Schwann cells. Need to be familiar with the name. In between each of these like little sections of myelin, you have this thing called a node of Ranvier. And node of Ranvier is, they're like these little gaps outside of the myelin sheath. And those parts are important for the next thing that we're going to talk about, which is what is saltatory transmission. Okay, so saltatory transmission is when nerve impulses jump from one node of Ranvier to the next. So basically the signals hop from one gap in the myelin sheath to another. So the first picture, and again, this is supposed to be a little GIF, but it's actually in the PowerPoint. So if you want to go back and look at that, the Canva PowerPoint, you could do that. Basically, the signal is going to move throughout this just uninterrupted. So it's just going to, but it takes a while to get all the way down if it's unmyelinated. When it's covered in myelin, what happens is the signal is going to jump from here to here to here, like that. And that jumping, that hopping, that's saltatory transmission. And it's caused by having the axons be myelinated, covered in myelin. Okay, depolarization and repolarization of neuron membranes. So this is kind of all about action potential and about how neurons fire and things like that. So depolarization and repolarization have to do with changing the charge. Of what's going on inside of a neuron. So depolarization is a change in the membrane potential from negative to positive. So we're starting from negative and it's going to positive. In response, this is how it happens, by the way. In response to sodium channels opening, that's really important. So sodium channels are going to open, and then there's going to be an influx of sodium ions, and that causes the membrane potential to become more positive. That's depolarization. Repolarization is the opposite process, which is where we restore the negative membrane potential. And this happens with the potassium channels that open. So potassium is going to move out. We call that efflux or effluxes, causing membrane potential to return to negative charge. So I'm going to show you a couple pictures, and hopefully that will help. I do also recommend there's 2 videos in the Canva slides that kind of go over this concept. I recommend that you watch those 2. They're pretty short. So here's a picture. It says propagation of a nerve impulse. So this is like when it's traveling. It says the direction of travel of action potential. So we're moving this way, which is the way that the nerve impulse is going to travel from this part down here called the dendrite. It's going to move out. So it says ion channels in the axon are voltage gated, which means that it requires an electrical signal or some sort of electrical work at play in order for the things to open and close. Depolarization at one axon segment triggers the opening of ion channels in the next. Consequently, the action potential spreads along the axon as a wave of depolarization. So if here's where we start right here with depolarization, and then it's kind of move as a wave. So as this depolarization moves that way, then repolarization happens, and then you get periods of resting, and it kind of happens over and over and over again. So think of it like if this is the depolarization that happens here, as it moves, this is going to return back to, it's going to repolarize, and then it's going to rest, and then it'll get depolarized again. Here's another picture that shows kind of what's happening with the sodium and potassium channels. So depolarization is electrical change where it goes from negative to positive. So the overall charge is supposed to be negative. So depolarization, D means like not or like opposite. So depolarization, it's undoing the negative charge. So it's going negative to positive. And what happens here is the sodium channel is going to open. We see that right here. And the potassium channel is going to stay closed. And the sodium-potassium pump is going to stay closed. And then repolarization, what happens is the sodium channel is closed and the sodium-potassium pump is closed, but the potassium channel is open. So in depolarization, it's going to pump sodium into the cell, and then you end up with that more positive charge. And then with the repolarization, it's going to pump potassium up out of the cell, and it's going to return it back to a negative charge. So depolarization, negative to positive, and repolarization is positive back to negative. Okay. All right, number 14, how are signals transmitted across a chemical synapse? So just so you remember, synapses are junctions, and junctions means like gaps or meeting place. Kind of like spaghetti junction where things meet. They're junctions between neurons and receptor cells. So neurons transmit signals across synapses by converting the electrical signal into a chemical signal. So this picture has like a 1 through 8, and it's kind of, it kind of goes into a pretty big detail, right? So we've got an action potential, which is some sort of signal, an electrical signal. And then what ends up happening is, so that's number 1, action potential arrives at the axon terminal. So the signal comes and gets there. Then number 2, voltage-gated calcium channels open. So calcium channels are going to open. Okay, and they're going to, then calcium is going to enter the presynaptic neuron. So presynaptic, that's this guy. Okay. And then the calcium is going to signal to the neurotransmitter vesicles. So that's these little synaptic vesicles right here. And the vesicles are going to move towards the membrane where they're going to dock onto the membrane. You don't need to know what a docking protein is. You just need to know that it kind of like sits there for a minute. And then it's going to, so essentially when it says dock, really what we mean is that it's going to fuse with the membrane. And then once it fuses with the membrane, all the stuff, the neurotransmitters are going to leave via exocytosis. So it kind of like fuses with the membrane and then pushes out all of the neurotransmitters. They're going to go through the little synaptic cleft, which is that like gap, where then the little neurotransmitters, the signals, are going to be received by the postsynaptic cell. So the shorter version of that, all those steps, is that the electrical signal arrives at the presynaptic terminal. The synaptic vesicles are going to fuse with the membrane. The neurotransmitters are going to diffuse across the cleft. And then the neurotransmitters bind to receptors on the postsynaptic membrane, and it produces some sort of electrical signals. Okay, so number 15 is to fill out the table below. And this table has all of the different hormones that we talked about last week about how we maintain homeostasis in the body. So let's start with insulin. Insulin is secreted by the beta cells of the pancreas. The target organ will be the liver. And the function of insulin is to decrease blood sugar. Now, insulin and glucagon, those are the ones that are kind of linked. So glucagon is secreted by alpha cells of the pancreas. The target organ is also the liver. And its function is to increase blood sugar. Thyroxine is produced and secreted by the thyroid. Thyroxine, thyroid. The target organ is actually all cells. There's not a particular organ necessarily that it's targeting every cell in your body. And the function of thyroxin is to increase body temperature. So remember, the presence of thyroxin means that your body temperature is going to increase. If it needs to decrease body temperature, your body will inhibit the production of thyroxin. Melatonin is secreted by the pineal gland. That's in your brain. And the target organ is the brain, specifically the pituitary. And the function is to regulate, and I think you guys know this one, circadian rhythm. Now, leptin, that's the last one, and this is another question later on the study guide, but it's secreted by adipose tissue, which is fat. Those are fat cells. And the target organ is the brain. And really, it's the hypothalamus, if you want to. As long as you know that it's a brain, that should be sufficient. And then the function is to regulate appetite. And really, when we say regulate, we mean suppress. Okay, and we'll talk more about leptin later on. Okay, number 16, what is the purpose of the hormones FSH and LH in the menstrual cycle? So FSH stands for follicle stimulating hormone, and it stimulates follicle growth and estrogen secretion in the menstrual cycle. And then LH, which is luteinizing hormone, it causes ovulation and the corpus luteum to form. The corpus luteum, by the way, is a mass of cells. It's formed by the ovary. Basically, if fertilization doesn't happen, then it degenerates over time. And if fertilization does happen and a person becomes pregnant, then the corpus luteum is kind of a mass of nutrients and things like that. Both chemicals, both LH and FSH, those are going to come from the pituitary gland. All right, so slides 30 through 32 on the Canva slides outline the process of the digestion of starch. Briefly outline the process below, including which organs and what chemicals they make. So it starts in the mouth with saliva. So we're outlining the process of the digestion of starch. So the saliva in your mouth contains amylase. And remember, anything that ends in ASE is an enzyme. So amylase, that's the enzyme that hydrolyzes starch into maltose. So maltose is one of the monomers of starch. It says then in the stomach, amylase stops working because the pH is so low, it's super acidic. So the starch kind of stays in this undigested form. Then what's going to happen is the pancreas, which is this guy right here. Okay. The pancreas is going to produce amylase. So the amylase is produced both with salivary glands and in the pancreas. The pancreas produces, I think, a lot more. So that amylase is going to be put into pancreatic juice. It gets sent to the small intestine through the duodenum or the duodenum. And this is where the amylase breaks down starch into maltose. Now, maltose, anything that ends in OSE, is a sugar. So then another enzyme comes through. It's called maltase, and that hydrolyzes the maltose into glucose. So starch eventually gets broken all the way down into glucose. And just so you know, maltase is produced by the lining of the small intestine. I would be familiar with this picture about where the pancreas is, and that the pancreas is responsible for creating that amylase, or most of the amylase that's used in the process. All right, number 18, label the structures of the human intestines and give a brief statement of what each structure does. So X is, these are villi, or the little finger-like projections that selectively absorb molecules. So that's what's for absorption of nutrients. Y, that's that first layer of muscle. It's circular, smooth muscle, kind of helps keep food moving. And Z is also smooth muscle, but it's longitudinal. And that also helps keep food moving. If you look at this picture over here, it's kind of one is a micrograph so you can see it, but it's in color. And the other one is just a little drawing, but it kind of helps give you an idea of what it looks like. All right, so it says HIV attacks certain cells in the body. Which cells are these? These are lymphocytes. Specifically, they're helper T lymphocytes. So here's a little screenshot. HIV stands for human immunodeficiency virus. It's a virus that spreads through the body fluids, and it affects those T cells or lymphocytes. And basically what it does is it makes the body sabotage itself and kill its own good cells. It damages the immune system. It replicates inside the human body. Of course, it has to have a healthy cell in the body to survive. And that's why it is highly transmissible. And it can be transmitted through body fluids and things like that. But lymphocytes is the big deal here. All right, so number 20 says, how does leptin control appetite? So we already kind of talked about leptin earlier on in the video, but basically what it is that your adipose tissue releases leptin. Then the target is the brain, so the hypothalamus. It inhibits appetite, and then by way of that, in theory, it will reduce your food intake. Now, just so you know, what happens though is that if you overload your system with too much food all the time, then your adipose cells stop getting triggered to produce leptin, and then you don't really feel full when you eat food. So that when your body doesn't produce leptin, you feel hungry all the time. You're going to eat more and more and more and more. So when the fat cells are empty, that's right here, no leptin signals, we eat. When the fat cells are full, the leptin signal is sent, it releases leptin, we feel full, we don't eat. This is how it's used to control appetite. All right, what are neonicotinoid pesticides and how do they interact with insects? Okay, so neonicotinoid pesticides, basically what they do, or just in general, neonicotinoids, irreversibly bind to nicotinic acetylcholine receptors and trigger a sustained response. That sustained response is permanent overstimulation of target cells. That's just a stimulation. So basically what it does is it prevents synapse/nerve transmissions. So what happens is insects have acetylcholine receptors that bind much more strongly to these neonicotinoids than we do, which means the pesticides that have neonicotinoids in it are highly toxic to insects and make it a super effective pesticide. So if you see in the first picture, normal neurotransmission, and what happens is the acetylcholine is going to go and bind, no big problem. But then when we have neonicotinoid, what happens is the acetylcholinesterase cannot break down the neonicotinoids. So basically it leads to paralysis. So paralysis/death. Specifically with these insects too. So that's why we use it as an insecticide, because the insects have, they bind more strongly in the receptors. So they're highly affected by it, kills them off by way of paralysis and death, because basically their neurons, their synapses are no longer able to fire. All right, let's outline how proteins are digested. We'll include the enzymes. So the enzyme that breaks down proteins is called protease. Another name for it is endopeptidase. If it ends in ASE, is an enzyme. Protease breaks down the bonds between amino acids, those are called peptide bonds, by adding water. That's hydrolysis. And then protein is converted to its monomer, amino acids. Okay. All right, why must the menstrual cycle be regulated in vitro fertilization? So a couple of reasons. Regulating the number of eggs produced can reduce the number of invasive treatments. Let's start there. So you take all this medication and in order to kind of hijack the menstrual cycle so that you can time when, the release of eggs and things like that, so that doctors can gather as many eggs as possible. The thing is that in vitro fertilization tends to be a very invasive process. And so when they control the menstrual cycle, they can kind of control and regulate the number of eggs that are produced. And then that way they can get more eggs in a cycle than they might if they didn't control it, right? It can also make harvesting those eggs easier. And it can help with the success rate of IVF by timing implantation correctly. So it has to happen at a certain time in the process. And when they can control it with medications, then they can time it more like exact. And then that will hopefully help out with the success rate. All right. Number 24. What is resting potential? What unit is it measured in, and how is the sodium-potassium pump involved? So resting potential is the difference in charge across a membrane when a neuron is not firing. Normally, resting potential inside neurons, which is measured in millivolts, you need to know that, is negative relative to the outside of the neuron. The sodium-potassium pump exchanges sodium and potassium across the membrane, and that should be a period right here. It expels 3 sodium for every 2 potassium ions that are admitted, and will also leak out of the cell. This should not say okay, well, this should say K plus will also leak out of the cell. This is going to create a gradient where the inside of the cell is more negative to the outside. So that sodium-potassium pump is going to kind of pump in and out sodium and potassium to create that gradient of charge so that the inside of the cell is more negative. And then if you remember this, I think it was in semester 1, the sodium-potassium pump requires ATP. It's something that it's an energy process. Which brings me to our next question. All right, so it says, what is the resting potential shown on this oscilloscope trace? So an oscilloscope kind of measures this activity. The resting potential is negative 70 millivolts, and I'm going to show you where it's at right here. So this little line in yellow that I just did, that right there is the resting potential. So it's right at negative 70 millivolts. You have to include the sign and the unit, please. That is something that they require on the IB exam. Okay, if you have any additional questions, make sure that you note them, send me an email, and I can pull together some more materials so that if you're having trouble understanding something, don't be afraid to also look in our textbook, which is posted on Google Classroom. Go back over our PowerPoint on Canva or you can just, you can send me an email, let me know what you need, and I can get it for you.